Guys, I am back and I hope, well, I took a break. Yeah, I hope by moving my camera I'm not upside down anymore. We'll see after I upload this. But anyways, moving on to lecture 4. Have a note on Christian Anfinsen and how he performed a classic experiment that demonstrated that the linear sequence of amino acids in a protein determines the folded structure of that protein. He used a chemical called urea and he gently unfolded, and this is highlighted, which protein? Ribonuclease A, and then reduced its internal disulfide bonds with mercaptoethanol. So the protein was ribonuclease A that he unfolded, and he unfolded it by denaturing it. We skip ahead and talk about catalysts and activation energy. One of the important functions of proteins is to function as catalysts for the many chemical reactions necessary for life. So know that. What is an important function of protein? To serve as a catalyst. So what exactly does a catalyst do? You guys already know this. We talked about it in workshop. It lowers the activation energy. Moving on to more highlights. What structure does DNA have? A double helix. What does the basic structure of a nucleotide have? There is a 5-carbon sugar, and it's either ribose or deoxyribose, right? And then there's a phosphate group, and then there's a nitrogenous base. So again, the 5-carbon sugar can either be ribose or deoxyribose. The nitrogenous base can be either a purine, which is CUT, cut like the pyramid, or so pyrimidine. Sorry, pyrimidine is cut, CUT, for the different nitrogenous bases, and then purines, A and G. So remember, uracil is a pyrimidine nitrogenous base, but it's not in DNA, it is in RNA. So, yeah. So the nitrogenous bases, he might ask a question on this, for RNA would be C and T, right? Not U, not uracil. I have highlighted the pictures for ribose, again, is for RNA. Deoxyribose is for DNA. We go from 5' to 3' for both ribose and deoxyribose. Yep. And so nucleotides, how they polymerize, and this was a highlighted note, the 3' hydroxyl attacks the alpha phosphate of incoming nucleotide triphosphate. Thus, polynucleotides are synthesized 5' to 3'. So I'll repeat. This is how nucleotides are polymerized. The 3' hydroxyl attacks the alpha phosphate of incoming nucleotide triphosphate. Thus, polynucleotides are synthesized from 5' to 3'. So which prime to which prime? 5 to 3. So we know DNA is a double helix, and when you look here, there are two sides to it, right? This is what we call anti-parallel to each other. One is 5 to 3, and the other is 3 to 5, so they're anti, but they are also parallel, so they are anti-parallel. And that's just what you call these black things. They are called the sugar phosphate backbone, not black gray. That was highlighted. Also notes on Rosalind Franklin. She was the one who did X-ray diffraction pattern. So know that, Rosalind Franklin, X-ray diffraction pattern. What did Crick do? Well, they took Rosie, they called her Rosie, which was very not nice, or that's not what she wanted to be called, but they called Rosie her Rosie, and they took Rosie's picture, and Crick knew that only a double helix would come from it. And so Watson started to play around for the model, and they finally put that together. Watson and Crick made the double helix model that we use for DNA today from Rosalind Franklin's X-ray diffraction pattern pictures. You'll need to know the percentages, so it's about 30% cytidine, sorry, it is, so you know base pairs, A goes with T, G goes with C, right? So Chargaff's rules say that the percentage of A equals T and the percentage of G equals C, right? Because they're base paired together. Thus, for example, if you knew that you had 30% guanine, if you had 30% guanine and you knew that it wasn't base paired with, say, A or T, then the percentage of A or T would be 20%. So again, when you have base pairs, A and T are together, and then G and C are together. So if G is 30%, then you know either C or so G, to explain better, G is always paired with C, A is always paired with T, right? So G is 30%. 30% times 2, right? Because they're base paired together, that's 60%. 40% is left. 40% divided by 2, if you wanted to find out A or T, both would be 20%. So, yeah. There is something called, for RNA structure, that I didn't really talk about because we were running out of time, I think, but RNA can loop back on itself and form a secondary stem loop. This is what it looks like, secondary stem loop. It essentially is looping back on itself to form internal self-based paired structures. And that's it for lecture 4. Moving on to lecture 5. So this is when we start talking about biological membranes, chapter 6. So membranes are, again, just going over highlights. Membranes are made of what? They are made of lipids and proteins, those two things. So let's just look at lipids first. What are the three types of lipids that we find in biological systems? We find 1, fats, 2, phospholipids, 3, cholesterol. No, there's 3. So when you're looking at fats and phospholipids, what is something that is common between the two? If you look at their structure, they actually both have a glycerol backbone. And that's important to know. So that's just introduction stuff, but let's look at fats. So fats are formed of 3 fatty acid molecules, or you could also say triglycerides, right? Triglycerides. There is an ester linkage in fats. So what kind of linkage? What kind of connection? There is an ester linkage. There is a hydrophilic head, and you need to know what it looks like, and then a hydrophobic tail. Fats are sometimes called triglycerides or triacylglycerol because it has 3 fatty acids and a glycerol, right? More highlights. Fats are not amphipathic. That's interesting. My notes are contradicting themselves. It says fats are not amphipathic, but right next to it, it says fatty acids are amphipathic. So I would actually go with the amphipathic one. Fatty acids are amphipathic molecules. This means that a water-soluble region and an insoluble region are found on the same molecule. Yeah. Phospholipids to form membranes are amphipathic as well. So what do fatty acids form? They form micelles. And so micelles, they're outside, there's the hydrophilic head. It likes water. Inside, you have the hydrophobic tail. It doesn't like water. So when fatty acids are added to water, they make micelles. These fatty acids function like detergent. Remember in workshop we talked about it. Detergent is soap, and it solubilizes fats and oils. So a question could be, how do soaps solubilize fat molecules? And the answer is when you wash your hands with soap, the greasy oil molecules and the dirt of your hands partition into the hydrophobic interior of soap micelles and are therefore solubilized and washed away. More notes. Some fatty acids are saturated, and saturated means they don't have any carbon-carbon double bonds. And these would be the ones that are linear are the ones that are solid at room temperature. Yeah, so saturated, linear, solid at room temperature. They're just really compact together. Other fatty acids are saturated, which mean they have exactly one carbon-carbon double bond, or they're polyunsaturated, which means they have two or more carbon-carbon double bonds. Unsaturated fatty acids, which make one carbon-carbon double bond, are always or most often, like really, really, really often, are cis in their transfiguration. And when they're cis, they are unfavorable. When they are cis, they have a kink. And because of that kink, they don't get to form as compactly as if they did not have kinks. So a kink makes it like this, kind of. Does that make sense? And so it's not as compact. And thus at room temperature, they're liquid in comparison to the saturated fatty acids, which are solid at room temperature. So what's an example of a polyunsaturated fatty acid? I didn't go over this in workshop. It is an essential fatty acid. And what does it mean by essential? Your body can't synthesize it, and you must get it through ingestion. More highlights. Examples of saturated fatty acids, remember these are the ones solid at room temperature, are butter and cheese, and these are typically animal fats. Again, they're straight because they can closely pack together to form a crystalline array. Even though it was a sidebar, I think there were some questions on trans fatty acids. So trans fatty acids, so a note on trans fatty acids. A partially hydrogenated vegetable oil in high heat, if it is solid at room temperature and contains trans fatty acids, what does this mean? Partial hydrogenation of oils leads to fats with trans fatty acids. There's a long process, but just know that's what happens. You have only some hydrogenation, but not complete hydrogenation of oils. And so those fats have trans fatty acids in them. Yeah. I think that's what my notes are trying to say here. Margarine is made by taking vegetable oil and converting their fatty acids with double bond kinks in them to these kind of fatty acids by hydrogenating them. Yeah, each of my slides have like a lot of notes in them, which is why I don't think they would be that beneficial if I sent all this stuff to you. But anyways, my notes say that trans fatty acids are favorable, or that gas, and you go from a carbon-carbon double bond to a carbon-carbon bond, there are no more kinks. So at room temperature, it will be solid. If you heat some trans fatty acid, you go from cis to trans. Oh, that's what it's saying. By going to trans, it is more favorable than staying in the cis position. So that's why people do give heat to vegetable oil. Know that HDL is good and LDL is bad. I have a note that says this stuff wasn't on the test, but I also have notes that this stuff was gone over in workshop. One sec, pause.